Here's our meiosis presentation by Payal Patel and Reema Chopra. Interphase. The chromosomes replicate during S phase but remain uncondensed. Each replicated chromosome consists of two genetically identical sister chromatids. Also, the centrosome replicates, forming two centrosomes. Prophase 1. This phase typically occupies more than 90% of the time required for meiosis. Chromosome also begins to condense, while homologous chromosomes loosely pair along their lengths, precisely aligned gene by gene. Metaphase 1. The pairs of homologous chromosomes in the form of tetrads are now arranged in the metaphase plate, with one chromosome of each pair facing each pole. Anaphase 1. The chromosome move toward poles guided by the spindle apparatus. Also, sister chromatids remain attached at the centromere and move as a single unit toward the same pole. Homologous chromosomes, each composed of two sister chromatids, move toward opposite poles. Telophase 1 and cytokinesis. At the beginning of telophase 1, each half of the cell has a complete haploid set of chromosomes, but each chromosome is still composed of two sister chromatids. Cytokinesis, the division of the cytoplasm, usually occurs simultaneously with telophase 1, forming two haploid daughter cells. In animal cells, a cleavage furrow forms, and in plant cells, a cell plate forms. In some, but not all species, the chromosomes decondense and the nuclear envelope and the nuclei reform. Prophase 2. A spindle apparatus forms here, and in late prophase 2, chromosomes, each still composed of two chromatids, move toward the metaphase 2 plate. Metaphase 2. The chromosomes are positioned on the metaphase plate as in mitosis. Because of crossing over in meiosis 1, the two sister chromatids of each chromosome are not genetically identical. Also, the kinetochores of sister chromatids are attached to microtubules extending from opposite poles. Anaphase 2. The centromeres of each chromosome finally separate, and the sister chromatids come apart. The sister chromatids of each chromosome now move as two individual chromosomes towards opposite poles. Telophase 2 and cytokinesis. In this process, nuclei form and the chromosomes begin decondensing and cytokinesis occurs. Also, the meiotic division of one parent cell produces four daughter cells, each with a haploid set of unreplicated chromosomes. Each of these four daughter cells is genetically distinct from the other daughter cells and from the parent cell. However, problems can occur during cytokinesis, such as non-disjunction. Non-disjunction is a failure of chromosome pairs to separate and can lead to several problems. Conditions such as Down syndrome, Turner syndrome, triple X syndrome, or Edwards syndrome can occur. Now it's time for some fun facts. How are mitosis and meiosis similar and different? One difference is in mitosis, two daughter cells are formed, and in meiosis, four daughter cells are formed because it occurs twice, meiosis 1 and 2. Another difference is that mitosis occurs in somatic cells, and meiosis occurs in reproductive cells. One similarity is that both go through PMAT stages. Another similarity is that both are types of cell division. How does meiosis contribute to genetic diversity? Daughter cells are a combination of both parent cell genes. During crossing over, new gene combinations are produced. Which meiotic division is most like mitosis and why? The answer is meiosis 2, because this is when sister chromosomes split, just like in mitosis. Also, in meiosis 1, crossing over occurs, which doesn't occur in mitosis. How many gametes can be produced, and how did you get this number? Our cell can produce 8 different gametes by independent assortment alone, because n equals 3, so 2 to the nth power equals 2 to the third power, which equals 8. And with that, that's the process of meiosis. Have a good day.